watching Science TV, and today's segment is on protein structures. Proteins are a macromolecule made up of one or more polypeptides. Wait, guys, I think I'm getting something. Breaking news! We just found out an amino acid is a monomer of polypeptides. Oh my god! And apparently, they're composed of a central carbon atom with an amino group, a carboxyl group, a hydrogen atom, and a unique R group. Proteins come in a wide variety of amino acid sequences, sizes, and 3D structures, which reflect their diverse roles in nearly all cellular functions. The four types of protein structures that we'll be discussing today are primary structure, secondary structure, tertiary structure, and quaternary structure. Now, off to Jane on primary structures.
The primary structure of a peptide or protein is the linear sequence of its amino acid structural units, and partly comprises its overall biomolecular structure. By convention, the primary structure of a protein is reported starting from the amino terminal end to the carboxyl terminal end. The sequence of amino acids in a polypeptide is stabilized by peptide bonds. Examples are amino acid chain of hemoglobin polypeptide. The R group affects a polypeptide's properties and function. The structure is hierarchical, which means all three are based on the primary structure.
Wow, Jane, that was some great information. Now off to Lydia on secondary structures.
Secondary structure is formed by a helix and a B-pleated sheets in a polypeptide. A helix is looped coils from hydrogen bonds that form between the oxygen of the carboxyl group and the amino group hydrogen of the amino acid. B-pleated sheets are folded patterns formed when two or more strands of a polypeptide are parallel, and hydrogen bonds form between adjacent and carboxyl, oxygen, and amino hydrogen atoms. Oxygen bonds form between peptide chains. Polypeptides must bend to allow the formation of hydrogen bonds. The secondary structure depends on the primary.
Thank you, Lydia. That was just lovely. Now to Siska on tertiary and quaternary structures.
Tertiary protein structure refers to the complete three-dimensional folding of a protein. This final shape is determined by a variety of bonding interactions between the side chains on the amino acids. As a result, bonding interactions between side chains may cause folds, bends, and loops in the protein chain. The different types of interactions are hydrogen bonds between polar R groups, ionic bonds between charged R groups, hydrophobic interactions between nonpolar R groups, and covalent bonds. The R group of amino acid. This bond is known as disulfide bond, and it acts as to stabilize the tertiary structure of those proteins that have such bonds. These structures can be altered by changes in temperature, pH level, and ionic strength. Quaternary protein structure refers to multiple polypeptides that intersect to form a single structure bonding two or more subunits. An example of a protein with quaternary structure is hemoglobin. In hemoglobin, one protein binds to oxygen, while another binds to carbon dioxide. This is how one protein can serve two functions. It also allows for a protein to undergo complicated conformational changes. This has several mechanisms. First, an individual subunit can change shape. In addition, the subunits can change their orientation with regard to one another. A multimeric protein can do either one of these, or it can do both.
You've now learned about the four protein structures. All four structures build off one another, starting with the primary structure, then to the secondary, then tertiary, and ending with the quaternary structure. Oh, a fun fact about proteins is that the lifespan of a protein is only 2 days or less. Another fun fact from an Oxford article by Leroux and Dick is that group contributions representing the 20 common amino acids plus 5-hydroxylysine and 4-hydroxyproline were used to compute the thermodynamic proteins of naturally occurring proteins that are found in a crystalline state in cells. One final fun fact is from the American Chemical Society by Pauling and Newman. The polypeptide chains are compatible not only with chemical and physical properties of proteins, but also with molecular structure. Thank you for watching Science TV. See you next week. Keep it classy, Oneonta.